Okay, hi. Welcome everyone to the second Malaria MOOC Hangout. We want to thank you guys for paying attention to this MOOC. And if you have any questions live, you can use the chat bar to send your questions, and Dr. Chitnis and Dr. Menard will try to answer them. And if we're going to try to answer all your questions, but actually, we're going to try to make today a bit more of a discussion about some relevant topics in the field. So I'm going to quickly go through one or two questions, and then we'll move on to the more discussion aspects. But feel free to send your messages live using the chat box. All right. So, and also I want to remind everyone that today is the last day of the Malaria MOOC. So if you haven't taken your final exams yet, or you haven't finished certain courses, today is your last day to view the videos. You can download the PDFs from the course, and you can keep those permanently, but you won't be able to view the videos any longer or take the questions or the exams. Okay, so let's get started. So one of the questions that came out after the second MOOC is about AMA1. And the question goes, since AMA1 participates in the moving junction formation during invasion, and it's secreted from the micronemes, and as it's described here as a surface protein, I was wondering if AMA1 remains expressed on the surface of the parasitophorous vacuole after this process. So I think Robert might take a go at this one.
Okay, thank you. Thank you for the question. So I think there are two keywords, or at least two ideas in this question. One is the fact that AMA1 participates to the tight junction or the moving junction, although I would add that the notion of the moving junction is probably not completely correct, because the junction itself is not moving, and it's the parasite that is going through the junction that itself is very stable. But one notion is the fact that AMA1 participate, which is obvious, but the question is still open to what it does in the junction. And the second, which is more the second part of the question, which is actually the center of the way it's phrased, it's where do we find AMA1, basically, and whether it remains expressed on the surface of the parasitophorous vacuole. So the answer to that question is no. So let's go back to exactly what AMA1 does during the invasion process. AMA1 is located in the micronemes and is supposed to be secreted at the anterior pole of the parasite at the time when the parasite actually hits the membrane of the host cell. It can be the merozoite or it can be the sporozoite. Then AMA1 associates with this very complex structure that we call the moving junction. It's quite tough to know exactly what AMA1 does in the junction. But what's absolutely sure is that AMA1 remains associated with the tight junction or with the moving junction during the entirety of the invasion process. Okay, so AMA1 is localized at the junction, as well as the posterior part of the entering parasite, which means that at the end of the entry process, AMA1, which remains localized at the tight junction, remains associated with the junction. Okay, so the parasite has entered into the parasitophorous vacuole, which is devoid of AMA1. AMA1 is not further secreted once the parasite has entered. And the AMA1 that was associated with the tight junction remains there. The tight junction remains at the plasma membrane once the parasite is inside the parasitophorous vacuole. Now, that's one part of the answer, which is basically where we see the localization of AMA1. The other part, which is a little more tricky, is what does it do? The general notion, which is shared by the majority of researchers, is that AMA1 is truly the junction itself, and that it makes the link between the actin in the host cell or whatever receptor in the host cell, and the actin in the parasite, in conjunction with proteins that are called the RONs. This model is substantiated by lots of data, including the presence of a complex between MA1 and the RONs. Other data, including our own, using knockouts of the parasite, both in Plasmodium and in toxo, have shown that when you knock out MA1, not just the parasite doesn't care, the parasite enters better, which is obviously a big problem to consider, that MA1 is really part of the junction on which the parasite pulls. So what I'm saying is that there are probably other essential functions of MA1 which would associate the two models, in which, sure, MA1 binds to the RONs, is located at the junction, but is not the junction itself, because otherwise one would not understand why, when you take it out, it's not that you don't have a complete phenotype, you have actually better entry. The last point, which obviously is sort of forgotten by those who want to sort of promote the classical model, is that in the absence of MA1, both in toxoplasma and with the merozoites of Plasmodium, there's a defect in adhesion before the formation of the junction, which is absolutely clear both for the merozoite and for the tachyzoite. So in my opinion, the only for sure and verified function of MA1 is a binding function to the host cell. Whether or not there's an additional function in the junction itself, I think remains totally open, probably to further experimentation.
Okay.
But just for the sake of the answer to that, to the question, the MA1 ends up not associated with the parasitophorus vacuole, but remains at the junction as it's left behind from the entering parasite.
Interesting. And do you think that could be targeted actually, since it's on the surface of a red blood cell that's infected, or would this just be not enough of the protein at that stage? I don't know.
So MA1 is not found on the surface of the infected red blood cells. You mean targeted on the surface of the merozoite?
The merozoite. So it makes this connection makes the moving junction, and then once it gets in, so where is it at the end of so it's still on the parasite?
Yeah, well, but it's obviously the half-life of what remains of the junction is extremely
little.
short.
Yes. Okay.
But if you stain, for example, MA1, you'll see something like 5 minutes after invasion, a dot of MA1 that corresponds to where the junction was.
Yeah. Okay.
And then 10 minutes after, it's obviously gone.
Gotcha. Okay.
So there's.
Okay, because I was trying to understand what the second part of this question meant about using serotherapy, but I don't think, maybe that's not related. The question of whether serotherapy is used to treat intense cases of severe malaria.
So the way I understand it, and Feta maybe has understood the question differently, I sort of made a complete difference with the first part of the question.
Yeah, I think you're right, yeah.
And has nothing to do with MA1. Now, whether antibodies as a whole have been used to treat severe malaria, the answer is no, not to my knowledge. And I'm not sure I would envision what the right application would be. Maybe to target the VAR genes, whatever, the determinants that are responsible for cytoadherence into deep vessels, which could be one sort of idea. The problem is twofold. One is that you don't know exactly the variant antigen that you ought to target because it's very changing. And as we know that there's antigenic variation, so whatever antibodies that you could start with, you don't know exactly how to continue. So I don't think that's a very valuable line of treatment. And as far as I know, this has not been used.
What about in terms of, I thought I read something about how you could take antibodies from an infected or an individual that was somewhat had already had an immune response, and then transfer them into individuals that had severe malaria. Maybe I just read about it as an idea and not as data.
No, those experiments have been done.
Sure.
They were done in the 60s, in fact.
Yeah. Okay.
The famous experiments of Cohen and later McGregor, where they took immunoglobulins isolated from sera of African immune adults and transferred them to children who were infected with falciparum, and that cleared their blood stage parasites.
Okay.
What exactly were the targets of those immunoglobulins is still not clear, and that is sort of the holy grail of malaria vaccine research.
Right.
It's clear that you can develop immunity to malaria over a period of time, and it appears it's not antibodies against any single or particular antigen, but a repertoire of antibodies against an array of antigens. And if we identify what those antigens are, one could then try to elicit that combination of antibodies using a multi-component vaccine. And that's something that we and many others are trying to work out.
Okay, that's interesting, because that actually brings us a little bit into the next couple of questions. But before we proceed, there's a question about, or we've been thinking about talking about a little bit about Vivax in Africa. So there was initially a question about this shifting between falciparum in some areas and then moving into Vivax. And this is quite intriguing in the case of Africa, where there hasn't been, or they hadn't detected Vivax in the past, and now there has been this increase in detection of Vivax. And I guess this is a question, this is quite an interesting area of the malaria epidemiology. So I don't know if you want to say a few words on that.
Yeah, this is a new, well, in the last few years, let's say, observation that has come up. And I think this will attract the attention of researchers over the next several years, which is the reports of Plasmodium vivax in sub-Saharan Africa. You know, the sort of textbook knowledge was that there is no Vivax in sub-Saharan Africa because the high rate of Duffy negativity, and Vivax needs the Duffy antigen to invade human reticulocytes, and that's why you don't have Vivax in sub-Saharan Africa. But as people have looked, they have actually found Vivax in sub-Saharan Africa and in communities where Duffy negativity is as high as 90 or 95%. There's also Vivax in Africa in places like Madagascar, where there's more of a mix of Duffy positive and Duffy negative people living in the same communities. But there also, this is the work of Didier Menard and colleagues from Institut Pasteur in Madagascar, where they showed the presence of Vivax in Duffy negative individuals. So it doesn't seem quite, you know, the sort of dogma that Vivax cannot infect Duffy negative individuals doesn't seem to is being questioned. Now, is that so? What's going? And then Vivax has been found in places like Mali, in Senegal, in Kenya, and local transmission is being reported. It's not just people coming from somewhere and who are Duffy positive in the community. It's actually local transmission in communities which are, you know, 90% or more Duffy negative, and there's local. But it's interesting that the levels of transmission are really low. For some reason, it has not it doesn't seem to go beyond a certain low level. And so one wonders, one needs to really work out the sort of epidemiology of this transmission, and then also at the molecular level, you know, what are the invasion pathways these parasites are using to infect Duffy negative individuals. There are reports, and this is not yet published but has been presented at conferences, suggesting that even in Duffy negative individuals, there may be a small window of time during erythropoiesis when early reticulocytes may transiently express the Duffy antigen. So actually, this is the stage where you may get infection of the parasite, of Vivax, into a person who's Duffy negative, but transiently expresses the Duffy antigen. in early reticulocytes. Alternatively, maybe something has changed. Maybe the parasite has developed an alternative mechanism to invade Duffy-negative red cells. And if that's the case, then one might see over a period of time the rise in Plasmodium vivax in Africa. And so one needs to keep an eye out for that, because you might, if that's the case, then it's something, vivax may be a emerging infection which you might see more and more of. And that is a worry.
Okay. Rob, anything, or can I?
No, I think
okay.
I think that's very complete.
Okay, sounds good. So I'm just going to shift tracks a bit from that area to get back to, to get to a bit of a history lesson, actually. But it's in line with our vaccine theme. So one of the questions that we got a couple of weeks ago, I think, was about the death of Dr. Ruth Nussenzweig, and the student was interested on maybe we could prepare maybe some sort of presentation or talk a little bit more about her research, and so basically. I was talking to Dr. Robert and Chetan about this, and so as some of you or a lot of you probably know, Dr. Nussenzweig made seminal contributions to the field of malaria, in particular the vaccine developmental area, and this was actually based on some seminal work in the 60s, where she discovered that attenuated or radiation-attenuated sporozoites could confer sterile immunity in rodents. And this was also the basis for a lot of vaccine, work for the past or till present. And then I just recently found out that actually Dr. Menard was incidentally a postdoc in the lab of Victor Nussenzweig, who is the husband was the husband of Dr. Ruth Nussenzweig, and he worked with him in the 50s, and I think also with Ruth, I believe. And so I think he knows a lot about her work, much more than me and Dr. Chitnis, and so he's going to give us some background on her contributions to this area. Yeah.
Yeah, I think the Nussenzweig couple is really among the two most important people in the malaria field. So obviously, I think it's worth talking of both of them, because I think it's impossible to talk about Ruth without talking about Victor, or the other way around. So, and what's interesting is that, or particularly fascinating, is that it's both a scientific and personal or family story, that of the Nussenzweig. So, first of all, Ruth actually was Austrian. I don't know if everybody knows, because usually people think that she's Brazilian, but she originally, she was Austrian, and actually fled out of Austria in '39 because she was non-religious, but she was a Jew, and actually left Austria in '39 and then arrived in Brazil. And did some MD studies. That's where she met Victor, who himself was Brazilian. And they worked together. They did some research in Brazil. And they were working in particular on the Chagas disease. They did an MD, they did a PhD, and then they went to NYU for a postdoc. And that was in 1963. You imagine. And during that postdoc, Ruth was working on malaria, and Victor at that time was working on some basic immunology stuff, and in particular the complement. He was working in the lab of Benacerraf, who had to have the Nobel Prize at that period, I believe. Now, another turning point in their life, where their personal story actually had a huge impact on their science, is that they both came back from the postdoc in '65 to find out a Brazil that had completely changed, because maybe some of you know that at that point there was a military coup in Brazil in '65, and so they went back to a Brazil that had completely changed, and what people also don't know is that Victor was a prominent political activist in Brazil. He was known to be a communist, which actually landing into some problem even in the US. And when he came back to Brazil, that was the Brazil of the colonels. And he actually told me that when he went back to whatever, it must have been the University of São Paulo, he was received by a colonel who told him, well, you know, from now on, that's what you're going to do. And Victor and Ruth took their luggage and went back to NYU and started their scientific life over there. So just to realize how the personal, geopolitical in some cases, and scientific life actually sort of intermingle. So when they were at NYU, Ruth was the only one who really started to work on malaria right away. Victor continued to work in particular on the complement and continued some work on Chagas disease. And it was in '67, so 2 years after they went back, that Ruth produced a main result, a main achievement, which obviously has put the field onto some very, very interesting rails. And that was a paper in Nature. It was the first demonstration that irradiated sporozoite in Plasmodium berghei, and it was a single injection at that time, was sufficient to protect against a challenge with Plasmodium berghei sporozoites. So it's basically, you know, the basis of the whole saga of the live attenuated parasites. So that came from NYU, from Ruth Nussensweig, who demonstrated the principle in rodents. Now, the little story is that she wanted to continue that work and to show that it was the same in humans, which was likely to be the case. And then because she was a woman, and because the only people onto which such a vaccine could be tested were prisoners, she was a woman, she could not enter into the prison, and therefore it's another first author that is the first author of the papers who demonstrate the same principle in humans. And that was in the early 70s. Now, from these achievements, Ruth has gone on, obviously working on the immunity to irradiated sporozoite, and she's been working at NYU, and she has directed the department, and obviously the countless contributions that they made in terms of understanding what was promoting such an efficient protection following the injection of irradiated parasites. Now that's on Ruth's side, and in parallel, Victor obviously made a huge contribution when he decided to completely focus on the malaria field, and he did that at the beginning of the eruption, if you will, of molecular biology in all fields, but in particular in parasitology, and that was the first opportunity to clone genes. And that was at the beginning of the 80s, and CS had been uncovered indirectly through monoclonal antibodies that were recognized as a major surface protein. And that's a science paper in 1980 that showed that you could protect mice by purely injecting monoclonal antibodies against CS, and you were protecting against the challenge with sporozoites. From that point on, Victor dedicated himself to the cloning of the surface antigen, which turned out to be CS, and the cloning was done in 83. I think it was done in 84 in falciparum, and from 84 to, no, it was in 83. In 84, the CS was basically given to GSK, which then continued the work and to produce the best delivery platform of the CS antigen, which turned out to be the fusion with the HBS antigen. But obviously, this sort of escaped Victor's hands. So it's just tremendous to see the impact that this couple has had on the field in terms of vaccine development, because they each on their side have been at the very, very roots of the two basic approaches to vaccinate today, which is the subunit vaccine, using the approach of presenting one antigen or maybe two, as opposed to present the immune system with an attenuated parasite, and the goal being obviously in this case that you hope to teach the immune system to recognize all, at least ideally, all proteins of the parasite, or at least the most important ones, which theoretically appears to be a more solid basis, obviously, if you immunize against more than one antigen, than if you just target one antigen, even if this antigen is by far the most represented on the surface of the sporozoid. Now, obviously, you know that the RTSS results are not that not that great, and they're certainly interesting, but the level of protection is about 50%, quite rapidly after the third immunization, and then the problem is that it wanes with time, and in a matter of a few years, the protection has gone. Okay, so that's
this actually brings us to another question. That should we stop with the? Are you okay with talking about Ruth, or should I?
Oh, I think I'm
finish up first.
I can.
You could talk much more.
I could talk, but unless there are specific questions, but otherwise, no, we can move on.
Oh, we can come back also, but I mean, this is interesting that you brought it up, because this whole question about the splitting of how they both worked on malaria, but these slightly different areas, which comes to another question about the use of a synthetic malaria vaccine. Someone asks whether it's not as good as, say, using the genetically attenuated, or I guess, which comes from this radiation attenuated approach. And so, there is this debate as to, maybe the gaps, or for example, or I don't know if there's a difference. Also, no, we could talk about the differences between the gap as well as the radiation approach in terms of immunity, and which ones appear to be promising, or the advantages and disadvantages of each of these.
Yeah, so there are basically two sub-questions. One is to compare the subunit and the live, and then the second is among the live, which of the irradiated as opposed to genetically modified would have the best prospects, right?
Right, and there's also this chemically attenuated approach, but that's, I guess, a bit...
I guess that's really... so chemically attenuated or arrested by a drug, because these are two different types. These are two different types. So you can either chemically attenuated the parasite, or you can have the wild type parasite.
Yeah, I meant the wild type.
That's right, because there are just a couple of papers on chemo attenuation, and I would not want to go into that because I think it's very, very premature.
Yeah, okay.
And I really don't think that's the sort of, the most promising. Yeah, but you're right. There's the third one, which is to block, to use a wild type, and then to block with
a drug.
So first, the subunit versus live, and obviously, Chetan, you can intervene whenever you want. Very schematically, I think that so we just said something important, which is that it's vaccination or immunization with a single antigen as opposed to the entirety of the repertoire. So obviously, if you look at things this way, then there's no contest. Why would you want to immunize with one antigen as opposed to many? Well, there are a few problems. First of all, we don't know how the various reaction to each individual antigen will mesh up. That's really something which is very empirical. And
I've could be diluting the response, for example, or...
So what we know is that yes, in fact, the irradiated parasite and the live attenuated parasites do protect. The antigens that are targeted are not known. We know that CS is also targeted by this approach, but what that counts in proportion of the entirety of the protection is unknown, and what are the other antigens that are recognized also is unknown. Now, the second level of reasoning when you compare the subunit and the live is obviously how easy they are to be used. So one thing is to have something that works more or less. The other thing is how efficiently can you use it on the field. And I think that's the problem of the live attenuated parasite, which is that they seem to work, well, better than some subunit vaccines. I don't want to say that they work immensely well in laboratory animals like rodents, but they work better. But at the same time, these are live parasites, which means that, well, then you have all of the problems that are associated with the means of delivery of such parasites. It means that they need to be transported in liquid nitrogen.
Reversion, I guess, also, if that's, or not reversion, but if any of them survive or aren't.
Well, that's another problem. You're absolutely right, which is that obviously the survival of the parasite in these vials is extremely low, and the last study that I've seen for that, which was inferred from the study, is that around 70% of the cryopreserved parasites are dead. So basically, when you re-inject a vial, probably less than 30% of the parasite are still alive. This may be one of the reasons why it's so difficult with the cryopreserved parasite to achieve great protection. But one can be optimistic and believe, and I think that will be the case, that we'll find better ways to cryopreserve and at the same time kill less the parasite.
Are you talking about the gap, or are you talking more in general?
No, I'm talking absolutely general.
Okay.
So whatever type of
any kind of live,
either being irradiated,
either
being genetically modified, or even the wild type that you will aim at stopping with a drug, which I think will not be a way to go in the field. Because
of resistance, or
exactly, I think because the number one reason is that it would be a fantastic way to actually, it would be a laboratory to select resistant parasites. And I think this will pose problems. And once again, I am not convinced that the people who are pursuing this line of research actually do it for the sake of commercializing that kind of approach, but we learn a lot about what protects in those conditions.
Right.
And in fact, the best results in terms of lasting protection came from these experiments from Bob Sauerwein in particular, who was the one really who was involved in that kind of research. Now, so I was talking about all of them. Well, you need to have something. Your parasite need to be cryopreserved, and you imagine that this is a huge problem for vaccinated in Africa, in sub-Saharan Africa, to the extent that many people believe that if indeed they really work well, which they seem to do, the future of these live attenuated parasites seems to be restricted to the West. So basically and this is ironical because, I mean, all this work has really been and is intended for a vaccine targeted, to the people who need it most. But for these technical considerations, one can very well imagine that we'll have centers in Boston, New York, I don't know, London, Paris, to vaccinate the Westerners who want to go to Africa, because this will be no problem to have these cryopreserved vials. And then it doesn't protect for a long time, so that's perfect. It might even protect military people. And military people are very interested in the live. But it seems more difficult to imagine that, and obviously things can improve, but at this point, it is a major limitation of this type of vaccination. It's really the very logistical consideration of how you use them in the field. In terms of how well they work, they work well. I'm talking about the live parasites. Yeah. And the major discovery that has been done in the recent years is that basically the later you block genetically the development of your immunizing parasite, the stronger the protection will be. Irradiated parasites are blocked midway through their development in the liver.
Okay.
And this gives a protection which is fairly good, but not fantastic.
Okay, so that would be an advantage of the genetically attenuated.
Complete advantage, and even more than that. I mean, I think because the last attempts that have been done to generate GAPs, we call them GAPs, genetically attenuated parasites, we're targeting a gene that is involved quite late in the development of the parasite.
But doesn't that also increase the chances of, for example, if there was like an escape, being closer to the blood stage would be a bit more risky?
That's a very interesting question, and obviously that's the major issue one should have in mind. I am not convinced that because you're closer to the end of the cycle, the problem is more. The thing is that not a single, you can have an escape at the level of the irradiation, and then it will continue. But you raised the very, very crucial issue, which is that another major problem of the of the live and the genetically attenuated will be to be sure that there's no breakthrough. Breakthrough parasite, meaning a parasite that has been genetically inactivated, but for a reason that is not clear, makes it through. And we know that some knockouts make it through the cycle. And the way to circumvent that will be to associate many mutations in order to have sort of insurances, if you will, that even if it escapes from one genetic block, then there's a second one and maybe a third one.
Right, sort of like making a multiple, multiple drug treatment, like multiple blockades. So I think I've seen up to 3.
Yes, the problem is that the 3 type of mutation gaps are, the 3 genes are very, very early. So it's basically a gap that is blocked very early and therefore doesn't have the best chance to be more protective than the rest.
Okay.
So in conclusion, the gap will be interesting if the genetic modification arrests the parasite very late in its development in the liver. That's point number 1. And number 2, if you associate the number of mutation it takes to be sure that by injecting millions of sporozoid, you're not going to have one that completes the cycle and that would infect a vaccinee.
Right.
Okay. So,
Ethan, any comments in that area?
Yes.
You work on protein vaccines too, so maybe some advantages of using that approach.
Yeah. So I think currently the jury is out as to which of the 2 approaches, irradiated or genetic attenuated parasites, so whole organism vaccines versus the subunit vaccines.
Right.
There are clearly advantages to each of them, but also challenges that need to be addressed for each of them. And I think Roberta has pointed out some of the challenges for the whole organism vaccines, the attenuated vaccines. With respect to the subunit vaccines, the leading vaccine is clearly the RTSS, based on the circumsporozoite protein, which, as we discussed earlier, was very early on worked on by Victor Nussenzweig and with GSK and Walter Reed, and then have brought to through clinical development, and currently is in a post phase 3 large implementation kind of trial where they're seeing how effective this vaccine will be. In the phase 3 trials, the results were mixed in that the efficacy was 30 to 50% against clinical disease, and only in children, not in very young infants. So we need to do better. So what are the challenges that subunit vaccines need to overcome to make better vaccines with higher efficacy? So clearly, first thing we will need to do is target multiple stages and make a multi-component vaccine. So if to say the CS, the circumsporozoite protein, you could add some blood stage components or another pre-erythrocytic stage component, then you could try and hope to improve efficacy. From all the analysis of what naturally acquired immunity does, it's clearly not against a single antigen, so you're not going to make a magic bullet. These are actually the development of antibody responses against a whole host of antigens, and we need to identify the key ones. The other challenge is, in addition to identifying the key antigens, is how do you make these antigens really immunogenic? Because for some reason, it appears that malaria antigens, when just produced as recombinant proteins mixed with adjuvants, are not very immunogenic when you don't get very robust immune responses, particularly in humans. So one needs to identify some delivery platforms, say like virus-like particles, hepatitis B fusions that were made for RTSS was one example, but I think there are now better platforms to make virus-like particles. That's one approach. Or one could make protein conjugates. That has been shown to also both boost antibody titers and make them more long-lasting, which is the other problem. How do you raise antibodies to high titers, but keep them high for a significant period of time, say at least a couple of years, or at least 1 year or 2 years, and then you can then come in with a booster. But typically, even with RTSS, the problem is the reason the efficacy is not high over, these are efficacies over 1 or 2 years, is because you actually have very high efficacy in the first few months, but then it drops off. And that's because antibody titers drop off. So what can we do to keep antibody titers high with our immunogens? And how do we take care of the polymorphisms in various antigens? Because there's great diversity in the parasite strains, the falciparum and vivax strains that cause malaria.
Genetic restriction also.
And on the human side, there may be genetic restrictions. So one can do that, address the problem of polymorphisms by focusing on functional parts of proteins, which are likely to be more conserved, and or focus on antigens that we know are conserved, or make a mix of antigens so that you cover a wide range of parasite strains. So there is a lot to be done, both on the side of the whole organism attenuated vaccines, as well as on the side of the subunit vaccines. And it's not yet clear to me, at least, which of the two are going to succeed. There's, I think, need for investments in both approaches. And I think hopefully, as more young people join the field and come up with new ideas, we'll hopefully solve the problem one day.
Right. It seems you can get you can get some ideas about the immune response using the genetically attenuated approach. So it's not necessarily just for the sake of making a vaccine, but also for more understanding of the...
for a basic understanding of what actually achieves.
What we don't fully understand exactly how, immunity is...
Absolutely.
Yeah, how the immune system responds to malaria, to Plasmodium. So actually, we're going to try to wrap up soon, and it's been very interesting talking to both of you and listening to getting a bit of a history lesson, which is quite interesting. And to a sort of lighter question right at the end, which is from a student that asks whether, Robert, you are related to Didier Menard.
So the answer is no.
Suspense.
No. So Ménard is a fairly common French name. I don't know what in India, maybe Krishna or
Patel.
or
in the
something like that. So and Didier and Robert are both common first names. So it's not that surprising. But we have nothing in common. Although we appreciate each other very much, Didier comes from the réseau, from the network, has been working a lot in Madagascar and in Cambodia for a long time, and I'm a very lab rodent malaria person. So no.
Different in both the.
So we're not from the same family and doing different type of research in malaria.
Right. Yeah. Okay. I would have guessed as much, but now we've answered someone's question as well. So thank you both so much for your time. If there's any comments you need to make right at the end before we say goodbye to everyone for good, feel free to do so now.
Well, I'll just say I hope we've certainly enjoyed putting this MOOC together, Robert and I, with all our colleagues who really came forward and gave their time and to do the lectures. And we've had very good participation of students. A large number have, I understand, followed the MOOC, and I hope have enjoyed it. And I think if we have sort of helped spread the word of malaria and the excitement in the field of malaria research a little, we would have accomplished what we started off to do. So thanks everyone for joining the discussion today and for following the MOOC.
Yes, thanks to everybody. I agree with Chetan. I hope that you will be the vehicle to spreading the words and the interest in malaria. That's very important. And I think we should also very much thank Aditi for her very fine work. So thank you to you as well.
Yes, thanks for seeing this MOOC through and being there to answer questions and everything through while the MOOC was on.
Thank you.
Thank you guys too, and thanks for all the students for paying attention and for your questions and comments and your introductions. Apparently this was one of the MOOCs, so people have been very friendly and introducing themselves, and this is not super common in the Pasteur's MOOCs before. So thanks for that and your positive attitude. And like I said, it ends today. I'm not exactly sure what time. I assume at midnight at Paris, but yeah, so take your exam before that. And if you have any questions, you better do so in the next couple of hours because it's going to be over soon. All right. Thank you, guys. I'm going to stop now.
Bye.
Thank you.
Bye.